Carl, welcome to the Center of Excellence in Biodiversity and Natural Resource Management weekly seminar. The seminar features research and projects that are being done by University of Rwanda students, University of Rwanda lecturers and researchers, and also from researchers and students from any other institution in Rwanda and internationally. And we really encourage anybody who may have a topic that they would like to present to please contact us. And Daniel is the one that organizes these seminars, and he'll help you get organized for this. It's a great opportunity if you have some research findings or you have a topic you would like to explore. We're very informal group, so you don't have to have something that is completely perfect, but it's a great place to get feedback and input. So today we're really, really happy to have one of the researchers at University of Rwanda in the College of Agriculture and Veterinary Medicine. I think I left animal health off in there somewhere. But we have Leonce Ngirinshuti. He's a master's student in our Biodiversity and Natural Resource Management program at University of Rwanda, and he's also in a dual program with the University of Koblenz-Landau in Biogeosciences. And he's going to be talking today about his master's research, which was exploring a particular group of insects, and he was working in Nyungwe National Park. And before we have him start, I just want to mention that what especially excites me about this research is that we can see we are developing taxonomic expertise here in Rwanda. We are developing expertise in different taxonomic groups that can eventually represent bioindicators or species that can tell us more about ecosystems. And Leonce is one of these people that's doing some very detailed work on a particular family, but he's also can use that to apply for understanding ecosystem functioning and health. And so we're just really happy to have him today to talk to us. So with that, I would like to pass it over to you, Leonce, and we're really looking forward to hearing you speak.
Thanks to everybody who attended the seminar. So as Professor Beth presented me, my name is Leonce Ngirinshuti, and I'm a student in biodiversity conservation and natural resource management at the University of Rwanda. This presentation is going to focus on a new species that we found in samples we collected in the Nyungwe National Park in 2018, and it has been published in the European Journal of Taxonomy. But before I proceed to the new species, I would like to give a brief introduction, a brief information about what Trichoptera is. Trichoptera is an order of insects that are commonly called caddisflies. And these insects are closely related to Lepidoptera. These are butterflies and moths. Their close relationship is basically supported by a silk gland that they possess in the head. And these glands is used to produce silk that the larvae use to construct the protective cases that they use when they're in water. Here it's an example of photos showing the larva that has grouped together different materials it collected from its surrounding using the silk. Trichoptera are also orometabolous insects. That is, they have a complete metamorphosis as it is the case in Lepidoptera. However, contrary to what Lepidoptera are, Trichoptera have aquatic larvae, whereas Lepidoptera have terrestrial larvae. That is why they are majorly considered as aquatic insects, because they spend most of their life cycle in water as larvae. Another difference between Trichoptera and Lepidoptera is that they have wing covered by hairs, whereas in Lepidoptera, they have wing covered by scales. Trichoptera, they are also used in biomonitoring studies as indicators of water pollution. So
Sorry,
our study, yes. Are you getting me, please?
Yeah, we hear great, but we're not seeing slides. Were you presenting slides, or is it just my system? I don't see slides. Daniel, are you seeing?
Yeah, I can see from my side.
Then maybe it's just from my side. Sorry, keep going. Yeah, I'll figure it out.
Okay, I think. Others, are you getting my slide, please?
Yeah.
Yes, I can see.
Yeah,
Okay,
you can
good.
keep going. Yeah.
Okay, great. So our study was conducted in the western part of Nyungwe National Park at different localities, including Chamudongo Forest, where we have had the 2 sites sampled. At Gisakura, we have had many sites sampled, and at few sites around Kamiranzovu and also in Uwinha. So we used the 2 types of traps for sampling. We used the Malaise trap. These Malaise trap, the insects get inside the Malaise trap through here. As they fly upwards, they got trapped into these bottles containing alcohol. The Malaise trap is mounted across a river or a stream, and it stayed there for a couple of days. The second type of trap that we used is the laetae trap. This laetae trap consists of a basin containing alcohol and a light lamp and a cover that prevents the rain from falling into the alcohol. And here are the batteries, and we have a white sheet of cloth. These help in amplifying light transmission. Contrary to the Malaise trap, the light trap are operated on night basis. This means that we mount it during the evening, and early morning we go and we remove it, and it is installed on the riverbank. Here is the list. This table shows us the list of rivers and streams that we sampled, as well as the number of specimens that we got per river. After collection, the sample were taken to the Swedish Museum of Natural History, where we conducted our lab activities. Our lab activities were partitioned into 4 stages. The first stages consisted in sorting the sample into different families. At this level, we used this stereomicroscopy, and we consider different morphological features like the length of antenna, like the patterns of wing venations, the presence or absence of simple eyes, like the structure of mouthpart, etc. The second activity was DNA extraction. With this activity, a section of an insect body was incubated in a solution containing this enzyme at an elevated temperature, about 56 degrees Celsius. Once the DNA is available in the solution, it has to be amplified. It has to be made into several copies so that it can be detected by the sequencing machine. So to amplify the DNA, we used the method of polymerase chain reaction, PCR, and we are targeting a section of the mitochondrial gene This section is called COI, or cytochrome C oxidase subunit, and it consists of 658 base pairs. For initiating the DNA synthesis or DNA replication, we use the two primers. Primers, they are just a short single-stranded nucleic or they may be also synthetic, and they are used to initiate or as the starting point of DNA synthesis. So once we have obtained the DNA sequences, they are introduced into the gene bank through an online platform. That is a screenshot of the online platform. Maybe I will play it later on. I can introduce one of the sequences that I have into the system and see what the result it is going to give. So the sequences are introduced to the gene bank to compare or to see if there are other similar sequences that exist there. This process of identifying species by using the short segment of their DNA is called DNA barcoding. So in our sample, we identified 9 families. Here it's the list of the families we identified. But as I had a limited time in Sweden, I only worked on one family, which is called Pisuliidae. I just choose to work on this family because it is endemic to Africa, and it is only found in mountain rainforest, including Nyungwe. Unfortunately, with this family, we only recorded 10 specimens during our sampling activities. So here it's the list of the recorded Pisuliidae specimen. We gave them codes, and they are the ones that I examined. However, in addition to the 10 Pisuliidae specimens, I also examined 2 more specimens from other families, and I'm going to explain why the 2 specimens were involved in our analysis later. So after the DNA sequences of the whole set of specimens was compared to the gene bank containing the database of sequences of the known species, and we found that only one specimen could be reliably identified as a species in this genus at a percentage of identification of 100%. The rest of specimens have had a low percentage of identification, so that we could not conclude that we have identified them by comparing them to the gene bank. For example, this specimen coded as RH1, it only got a percentage of identification of 887 as Silvatares and Sifa. And this percentage is very low to conclude that it is this species. However, it's also early to conclude that they are new species, because the gene bank seems to be incomplete. It seems that all known species don't have their sequences available in the online system. That is why we proceeded to the description of the species, to the morphological description of the specimen, and compare them to the description that had been done before previously by other researchers. again, as my time was limited in Sweden, I only worked on the description of one specimen, which is coded as RG8. So this is the picture of the specimen I worked on, and this picture was taken on when the specimen was still alive. The specimen was named Silvatares laetae, and laetae means happy, and it was named after Letitia Nyinawamiza just for recognition of her support to many students, including myself, who were interested in aquatic studies. So this species, we collected the 2 individuals of this specimen in our sample, and the one we worked on is holotype. The others that we, not the one we used, we examined for a description, is called the paratype. Here, the text here, it's the information about the holotype and the paratype, just to show the country from which the specimen was collected, the locality, the river from which it has been collected, the geographical coordinates, the date of collections, the people who collected it, the name of the type of trap we used, the vials in which the DNA is conserved, as well as the museum at which the specimen is conserved. So here we have a photo showing us the drawings of the wings and the genitalia of this specimen. Actually, in insect, we use male genitalia for describing the specimen. The genitalia consist of the posterior part of insects, including segment 9, segment 10, as well as other associated structures such as this one, which are called inferior appendages, and the penis. Penis is also called the phallus. After examining the genitalia of Silvatares laetae, we found that the genitalia, they resemble the genitalia of another species that was described from Uganda, which is called Silvatares exersus. So the similarities are based on the shape of or the structure of tergum 10, as you can see on both species. The tergum 10, it consists of a robe that is curving downwards in both species. Another similarity, it is these inferior appendages when they are observed in ventral view. The inferior appendages are curving inwards in both species. However, although there is some resemblance between the genitalia of both species, the new species was distinguished from Silvatares exersus by this mouth-like structure. It's a mouth-like depression that is at the tip of tergum 10 that is not observed in Silvatares exersus. Another difference is a pair of triangular lateral structures that are projecting from the tergum 9. That is tergum 9 observed in dorsal view. Here it is observed in lateral view. And you can see that these structures, these lateral triangular structures, they are not observed in Silvatares exersus when observed in ventral view or even dorsal view. Another difference was based on the structure on a pair of two processes that stand erect at the tip of the phallus or the penis, when it is observed in ventral view, they are standing erect, whereas here they are laterally oriented. So basing on this difference, we found it as a new species. Another difference that has been was observed on their wings. Here, on the underside of forewing of Silvatares laetae, we have large and long golden hairs that are not observed on the wings of Silvatares excelsus. And here we have I just showed the length of the wings, both forewings and hindwings wings of Silvatares laetae. So we also performed, or we also tried to establish a phylogenetic relationship between the specimen that we analyzed of the family Pisuliidae, together with the specimen of the family Pisuliidae, for which we were able to find their barcode or their DNA sequences. As I told you, we also analyzed 2 specimens from families other than Pisuliidae, and I have told you, I will explain why we included them in our analysis. We just included them in our analysis so that they will help us to root the tree, this phylogenetic tree. After the analysis that we performed by analyzing the DNA, the genetic distances existing between the specimen that were involved in the construction of this phylogenetic tree, we found that these 6 specimens are probably of the same species, together with another species that was described from Madagascar. We also found that the 2 specimens, according to the genetic distance existing between them, which is below 3%, they are also of the same species. And the 2 specimens are probably of the same species. The one we analyzed as holotype, it is this one, coded as RG8, and the paratype is RH9. And this analysis also showed that they belong to the same species. We used this software for constructing this phylogenetic tree. So this is the end of my presentation. I just ended it with a picture of myself, my supervisor, and the park rangers who helped us to reach different sites from which we took our sample. I thank you very much for your attention.
Thank you very much, Leonce. Beth is not around. She's joining another meeting, so I may take over. So I would like to ask anyone with a question or comment to ask. Feel free to ask. You can interrupt, you can write in the chat, or you can unmute yourself and ask.
Can I stop sharing so that people can?
No problem.
Okay.
Anyone with a question, comment?
Yes.
Yes, Joseph.
Well, thank you for the presentation. Actually, I see it more elaborated, and I'm very bad at it actually. So I would like to know if we have any plan to go further with the other specimen you've collected? And also, how do you preserve them for the time being? Thank you.
Okay, thank you, Joseph. Actually, the plan was to continue examining the specimen that are stored in the Swedish Museum of Natural History, and I would have returned to Sweden this year, but due to this pandemic, it has been postponed. And for more information, we wish to continue our research in different ecosystems, Rwanda, especially these mountain forests, where we think there are many species which have not yet been described. Then your second question, can you remind me the second question?
Actually, it was by curiosity asking you how you preserve the specimen.
Okay. Yeah, actually, the specimen are preserved in a concentrated alcohol, pure alcohol, and they are also conserved at a very low temperature, below, in minus 80 degrees Celsius, and they should be preserved in a museum. It's actually one of the guidelines, or it's one of the obligation when that is given by international. It is the International Zoological Code of Nomenclature. The specimen should be conserved in the museum so that they can be accessed by any researcher who wants to explore or who wants to contrast what you have done. Thank you very much.
Great. We have another question in the chat from Zara Alphonse, who says, thank you for your presentation. Can you share with us exactly the location of the new discovered species?
The location of the new species?
Yes.
Okay. Maybe on the map is not going to be clearly observed because I failed to include the river or streams in the map because I didn't have the shapefile containing the coordinates, the geographical coordinate of the rivers, but it is in Gisakura. It was collected from Karamba River, which is in this area, in this zone.
Okay, I hope he can notice. And we have another question from Tasia Hagenmana, who is asking, I would like to know the role of Trichopterans. Is there any mutualistic or antagonistic relationship between plants and Trichopterans?
I don't think there are any mutual relationship between Trichoptera and plants. However, the Trichoptera, they are benefiting from the vegetation that are nearby the river or the streams or any water aquatic body, in which they exist, because the Trichoptera, they prevent the sight from people or other animals. So they are hiding, they spend the whole daytime hiding in the vegetation which are near the river or near a water body. And so they are eating from the vegetation. Also, they feed. When they are adults, they feed on nectar. They spend a small time as adults, but when they don't feed, when they're adults, they only feed on nectar. So I think they can benefit from plants, but I don't know benefiting as a mutualism. They are benefiting the plant. That is the answer that I can give.
Yes, he also asked about its specific role, I think, in ecological role.
Their main ecologically, they have a great role because they serve as food for fish and the other living animals that are in water. Ecologically, also, they help to remove the moss that may grow into water because they feed. These larvae, they feed on other larvae of other insects, although some are herbivorous. Also, these the larvae of Trichoptera, they are importantly used in biomonitoring studies. I had mentioned it because they serve as excellent indicators of water pollution and aquatic environmental health in general.
Okay. While we wait for more questions to come in, I would like to ask a curious question. You mentioned about going to Sweden for this research. I was wondering if there is no lab in Rwanda that can help to identify a new species, and if not, what do you think is the way forward so that we can have that ability in Rwanda?
Yeah, I think it's not because we don't have a laboratory that can be used for barcoding, but the process, because my supervisor was from Sweden and I was linked to him, so that is why I was also associated with his project, and he came to take sample and bring them back to Sweden. However, we also don't have ways of conserving the specimen in our museum. I think we have one museum in Karongi that is starting to be associated with environmental and research activities, but they have not yet started having ways of conserving the specimen that are discovered or that are decoded from Rwanda. So I hope as long as we sorry?
Yeah, just because this is such an interesting discussion here, that Daniel's question and your response. And this is what the Center of Excellence in Biodiversity and Natural Resource Management is doing. And we hope that the legacy for the center will be to bring together researchers like you, Leonce, who know how to collect different specimens. In particular, as you indicated, like caddisflies could be important indicators, and then have a way to preserve those specimens and maintain them for research and teaching, etc. And really invite you to come work with a team that is putting together the zoological collections as a sister unit to the National Herbarium, which of course are the plant specimens, and they are being digitized and made available online, and they can have metadata about where they were collected and other kinds of information. But also your data would be really valuable for the biodiversity information system, where eventually there could be using caddisflies to look at their distribution in relationship to land use and the different qualities of different wetlands. So I think that question Daniel asked is really great. We're starting to work more with the biotechnology collectible conservation genomics lab and the capacity to do that. So we really need your kind of expertise to get that going.
Yeah, sure. Thank you very much. I will be pleased to be part of the team. will be involved in the conservation of our specimen.
That's wonderful. It's so impressive what you did here.
Okay, thank you.
Thank you both. We still have a question from Joseph. He says, I know Jean Dodier Wieselman is working on Lepidoptera in lab in Cumberland's, London. Any plan to join your effort with him?
Sorry, any plan? Can you repeat again?
He's wondering if you plan to join efforts with him for his research.
Yeah, sure. We have been collaborating with Jean Dodier for his research, although I'm not yet involved in his research, but we talk, we share information, and I'm aware of the research he's doing, on Lepidoptera.
That's great.
Okay.
Sure, go ahead.
I have a question.
Yeah.
Okay, thank you, Leonce, for your presentation. I have a small.
Sorry, your voice has
indicator that species can show us.
Sorry, can you repeat again the question? Yeah, sorry.
Yeah, maybe.
So you said about the I don't know exactly the species, but you said that it is an indicator of pollution. In water. What kind of pollution there?
Okay, thank you very much. Actually, it is not this species that is only an indicator, and they have not studied whether it can be used as an indicator or not. But in general, Trichoptera, because they exist almost in all aquatic environments, and they have a large diversity, and many of them have been used as indicator of water pollution. So the type of pollution depend on the type of species that is being studied. For example, whether it's pollution related to agriculture runoff, whether it's pollution related to industrial waste, any kind of pollution, depending on species that has been found in that area. Generally, they are considered, they are appreciated as good indicators of water pollution. It's not the only group of insects that are used as indicators. We have also dragonflies, we have mayflies, we have many groups of insects that are very important indicators of water pollution.
Okay, now I have another things about the use of molecular markers in taxonomy. So what kind of body did you use when you were extracting DNA?
Okay, we used abdomen. Remove the abdomen, and the abdomen was incubated. A solution that contains an enzyme, and this enzyme will destroy the cells and have the DNA now available in the solution. So used it's only the abdomen that is used. Any part of an insect body that you use, it has this kind, it can function well for DNA extraction.
Okay, now, okay, something that I want to So you use an automatic machine for extraction, or did you use like a manual methods of extraction of DNA?
No, we used an automatic machine. We just prepared our solution and we gave it to the machine, and the machine handled the rest of the process itself for DNA extraction.
Okay.
Yeah.
Thank you so much. Yeah.
Thank you very much for your question.
Thank you. Just another curious question. Do you plan to do research about IUCN threat status of this new species?
Not really. If there is no such plan, actually in Trichoptera, we only have 3 species that are threatened, and the rest are not really threatened, or they are not in danger. So I think that I have that kind of plan. Our nearest plan is to do like an inventory in different ecosystems. They have like a catalog of all the existing Trichoptera species in Rwanda. Yeah.
Okay, that's great.
Yeah.
So it's nearly time. So if there is no
sorry.
Yeah.
That you kept the sample in Sweden. Eh?
Yeah.
Why you didn't bring another one in Rwanda? Even you give the name of the 4 principle. You give the name of that. Yes.
Yeah. Actually, I was associated to the project as a student, but I was not really the head of the project. The head of the project is a researcher from the Swedish Museum of Natural History. And maybe, so I think through through further work that we'll be doing, we can have also an institution like Professor Beth said, we can have our Center Excellence developed at the level of hosting all the specimens that we'll be able to collect in Rwanda in the future. So that is why at the moment the sample have remained in Sweden.
Okay, I hope that answers the question. So
yeah,
if you don't have more questions, we can conclude this seminar. So I think there is no more question. So let me take this time to thank you, Leonce, for giving this seminar.
Yeah.
And I hope everyone will keep joining our seminars every Wednesday. Thank you very much for your attendance. Bye-bye.
Thank you very much.